I'm very much interested in the way different bacterial cells from different strains or different species interact with each other in the context of forming biofilms within three-dimensional environments. So very much interested in understanding how the biochemical and biomechanical composition of the three-dimensional environment where bacteria could be embedded or found would affect the way they interact with each other, the way they cross-talk, and the way they form biofilms, the way they form resistance in biofilms, and how those biofilms respond to novel processing approaches like plasma, for example. There's a great challenge and that applies not only in the food industry, but generally globally, and that's antimicrobial resistance development. So we know that bacteria, including bacteria that could be found from farm to fork in the food chain, are becoming incredibly resistant to the environment, but also to processing conditions. And as a matter of fact, according to the O'Neill report, by 2050, there will be more people dying from bacterial infections than cancer, which gives you an indication of how important the problem is. And how does antimicrobial resistance link to our research? Well, bacteria, when they communicate, they do transfer resistant genes from one to another, and they develop antimicrobial resistance on the one hand. And on the other hand, when you treat them with milder processing approaches as compared to the conventional approaches, this mild treatment could lead to the progressive adaptation of the bacteria to the treatment and eventual development of antimicrobial resistance. So there's definitely the practical consumer-driven demand, but there's also a global challenge that we need to face, and that's the AMR development.